I'd like to introduce our next session here about deep sea protection. I'm going to introduce Andy, who's our Director of Science and Operations from our Eureka office, and Peter Etnoyer, Dr. Peter Etnoyer, who is a deep sea biologist specializing in the taxonomy and ecology of deep sea corals. He serves as the lead for the Deep Sea Coral Ecology Lab at NOAA's Centers for Coastals and Ocean Sciences. He also serves as an adjunct professor at College of Charleston and conducts benthic surveys of deep sea habitats throughout, basically everywhere, the Northeast Pacific, Gulf of Mexico, Caribbean, and Atlantic. And he's going to be talking about some of his work around the country. And if Andy and Peter can join us, that would be great. Thank you, Andy and Peter. And don't forget to unmute and take it away, Andy.
All right. Thank you, Natasha. Hey, Peter. So thank you for joining us here today. Excited to have this little discussion about deep sea corals and the work that we've been doing. Obviously, you know, we've been working together since about 2012. It's been just an amazing learning process for me in terms of knowing what a deep sea coral was. I've always had an understanding what shallow water corals were, but you know, what a deep sea coral is. And I think today discussing that will be kind of a unique experience for everyone. So thank you again for joining us.
Sure.
So let's just, yeah. So let's just kick this off. Can you just tell us about deep sea corals? Like I said, you know, what is a deep sea coral and why are they important?
Sure enough. You know, I could take two approaches to this question. I could either talk about it scientifically, or I could talk to you about, you know, how the fishermen see deep sea corals. And they were some of the first ones to encounter these animals in the deep water. As some people might know, you know, trawling, deep sea cables have been around for 100 years or so. And when they pulled up these cables from the deep sea, or when they pulled up the trawl nets, every once in a while, they'd find things that looked very familiar, but wouldn't be expected down there in the deep sea. And so deep sea corals are referred to by the fishermen as the trees of the seas. And I've always kind of liked this expression because they do have very similar ecosystem function. You know, they can grow pretty large in the deep sea. Pretty large is a meter and a half tall, not like the trees we have on land, but they're often have fish hiding out in the branches. They have shrimps on them, crabs on them, brittle stars. So there's a whole ecosystem that revolves around these branching structures. And they provide the same kind of structure and refuge to the animals in the deep sea that, you know, the tree branches provide to fishes and squirrels and stuff on land. So they're important ecologically. Scientifically, they're very similar to the ones you see in a scuba diving magazine or a snorkeling magazine. Pretty much all the different kind of corals that you can see in the shallow water, the scleractinian stony corals, the sea fans, and the black corals, they also exist down in the deep sea, but they're adapted, of course, to a very different type of environment. The temperatures down there are typically between like 4 and 10 degrees Celsius. So about as cold as your refrigerator. And they don't get light down there. It's pretty dark. So they rely instead on the particulate organic matter, or the plankton that rains down from the surface. So this is a key difference between the deep sea corals and the shallow water corals. The shallow ones have the symbiotic zooxanthellae. The deep ones have a whole microbial assemblage that we're still learning about. But we know that even though they exist in the deep sea down to 8,000 meters, actually, there is this connection to the surface environment. And that's important to understand for their health and also just to predict where they might occur.
Well, and you described essentially, you know, sort of the forests of the seafloor, if you will, you know, the short trees, Joshua trees, or however you want to describe them. But, you know, as their shallow water cousins would go, how do they grow? I mean, the growth rates, I imagine when it's this cold, dark, and deprived of some of the nutrients and sunlight that you would expect in the shallows, I imagine growth rates then are incredibly slow as well.
Yes, they can be very slow, and we're still trying to get a handle on this. You know, the best way to figure it out is to actually collect a sample, take it back to the lab, and do all kinds of radiochemistry and stuff. What we've learned is that just like the shallow corals, many of them are decades to hundreds of years old, and some colonies have even proven to be literally thousands of years old. So as far as I can tell, they both grow slowly, but different types of corals can grow actually pretty quickly. So you got to know which species you're working with. But yeah, they do grow on the order of centimeters per year at their fastest.
Oh, well, that's actually quite a bit then. Okay. Well, so now that we understand, the difference kind of between a deep sea coral and the shallow water corals, what we're seeing changes then in these communities based on what we're seeing in the changing ocean, whether that be climate change, warming, that kind of a thing. Are you seeing any of that, or how's that going?
Yeah, well, this has been a question as a marine biologist and a conservation biologist. I consider myself a conservation biologist as well. It's always been important to me to understand whether marine protected areas can really protect corals from all the challenges they face. And climate is definitely one of those where we work together in Channel Islands and Southern California. We have a really kind of a unique and dynamic environment. We've had the warm water heat waves that we've found go quite deep. They can go to 100, 150 meters for a couple days, not like they do in the surface where they last for weeks and months. But also, there's the threat of ocean acidification, which comes from more and more CO2 in the atmosphere. It's absorbed into the oceans, and it's also transported in the Atlantic very quickly at the poles down to the deep sea. So once again, although we think of the deep sea as this kind of isolated environment, there are these rapid connections between the surface and the deep sea, depending on where you are in the world, of course. Pacific, as we've learned, is one of those areas where there is no ventilation. There's no water sinking at the poles. It's an acidified ocean at the end of the global conveyor belt. So we have found that the corals growing there in this acidified environment present themselves very differently than the same species growing in the Atlantic, where the levels of aragonite saturation are higher, and the pH is also lower. And there they'll thrive and form hills and mounds that go on for kilometers. But what we've seen in California, that same species will grow to be, barely the size of your hand. We didn't even know it was the same species until we were able to collect this using the beagle and get some DNA that confirmed, yeah, you have the same species here, but one is growing in an acidified environment and the other one is growing in a more basic environment.
You're talking, this would be the Lophelia species then, right? And I think you had a paper out that kind of sort of coined a term for that sort of loss. I think it was coral porosis or?
Coralloporosis, they were calling it. A lot like the osteoporosis that can affect older people. When you have, reduced calcium and increased pH, you can get more porosity in the skeleton. And that porosity makes it very fragile, easy to break. And this turns out, this is what we think, that's the reason we don't get these reefs forming in California. They just naturally form these smaller clusters, put more energy into feeding maybe than they do building their skeletons. Whereas out here on the Atlantic, it's a very different story. They have enough strength in those branches to just keep growing on top of each other till you get literally, a large mound or a hill of these branching stony corals.
Well, in your experience, so we see these large mounds of corals, and as far as richness and diversity, are we seeing that these tend to be spots where other organisms also aggregate and become sort of, I guess, hotspots for sort of biodiversity?
Absolutely, absolutely. That's a good description for it. They create so many little nooks and crannies that it just provides a lot of refuge for the invertebrates that probably serve as a food source to the mobile fishes that are down there. And also, you know, we see some types of cat sharks laying their eggs on these corals. We found that squid and octopus can also lay their eggs on these corals. It gets them up off the seafloor, away from the predators. Makes it a little hard to get at, but definitely hotspots of biodiversity, and more. And at NOAA, we're using deep sea corals as kind of a proxy for biodiversity in the deep sea. A lot of work has been done on the sand and the infaunal creatures, the little worms and urchins and gastropods and things that can live in those sediments. But we're just starting to learn about the hard bottom habitats and the types of animals that can grow on those. But where we find those hard bottom habitats, we often find corals growing there and sponges as well. And these, you know, form that important structure for all the associated animals.
Well, and I imagine that these habitats then are very, you know, obviously being sensitive to the acidification, but then when you think about some of the fishing pressures that might be put upon them, if it's trawling or even just some you know, anchors could be quite devastating into these habitats.
That's right. That's right. You know, we used to think of bottom trawling as the only major threat, and we have seen examples of this in the Atlantic. NOAA writ large has been studying oculina reefs, which are down there around 50 to 150 meters off the Atlantic, and this is a hotspot for fishing. Right?
Okay, right.
The depth zone is right. The Gulf Stream is there. Yeah. But when they trawled over it, you know, they went back, the people who did this work, and the area is flattened. So there's no more structure. There's no more branches for the animals to hide in. No more base of the food web for these mobile predators. So yeah, bottom trawling is, we consider it a destructive type of fishing. Now, the fishermen have found ways to use these nets more responsibly, right? They have a lot of control. Just knowing that the corals are down there has been very useful. We find the fishermen, they understand these habitats are important to their target species. So we've had a lot of support actually from the fishermen to protect these kind of habitats where they occur. Definitely the bottom trawlers, you know, they are not looking for rocky, complex habitat.
No.
Nice, sandy,
very expensive nets. Yeah.
That's right. Very expensive nets.
Yeah, but I know when we've been out before, we've seen, you know, even like long lines that will get themselves wrapped around rocks and choke to point where it rips things off. And so I know it all has sort of an impact, but you know, as far as the work that you're doing, we're seeing the management and fisheries responses are starting to merge together, where management decisions are made based on areas of high biodiversity hotspots, if you will, and fishermen now are beginning to stay out of those areas, understanding the value and importance of them. And
that's right. And it's all types of fishing, not just the longliners and the trawlers, but pots and traps, you know, for sablefish, which are very deep occurring fish. And we observe those in these habitats. They have a very small footprint, right? But over the years, if you're going back to the same spot over and over again, it could do some damage. But, you know, I don't know if a lot of people know this, but only, 12 years ago, they revised the Magnuson-Stevens Sustainable Fisheries Conservation Act to include deep sea corals as what we call essential fish habitat. So before that time, the fishing management councils didn't have this tool in order to, you know, delineate these habitats and limit the types of fishing that occur there. And, you know, at NOAA, we support fisheries. We support sustainable fisheries. So we want people to be able to access these habitats, but be able to do so responsibly without leaving their gear behind.
No, exactly. Well, then that's maybe a little great segue to go into sort of the coral work that we've been doing around the Channel Islands, you know, in partnership with you and the sanctuary, looking at that. So you want to just tell us about that work and sort of a subset of what we've been doing?
Yeah, sure enough. So Channel Islands National Marine Sanctuary is about one of a dozen sanctuaries we have around the country. It's situated right at the intersection of two major currents, the California current, which is coming down from the north, and the Davidson current is coming up from the south. And so it turns out to be one of these biological hotspots that's driven by the oceanography, but also by the broad, shallow shelf around the Channel Islands and the islands that stick up out of the water there. Down below, we have canyons, mounds, ridges, all these features that we're just starting to discover. Although the sanctuary's been around like 20 years or so, we're still mapping. It's hard to map, especially in these kind of shallow intermediate waters. But where we do get that work done, we're seeing more rocky features and learning how to access them and survey them in a repeatable fashion. And when we go down and when we look in the 50, 150 meter zone, where there's a rocky ridge, there's going to be some kind of, you know, epifauna, whether it's corals or sponges, but also, you know, the abundant and beautiful rockfish that occur there. And those are often found with the corals, but they're also found without the corals. So we don't understand the relationship yet between the fish and the corals, but we know the corals grow slowly. They're hard to replace. They have a value in their own, and that they're very common and conspicuous down there in the deep waters. And so the work we've been doing is to map these out very finely, right? So we match the biology data to our geology data, and we can tell you exactly how many corals were observed on any dive and how big they were and what species there were. But what's really cool about the work is that we can carry it forward with habitat suitability models, right? So where we say, hey, when we saw this at this depth pointing in this direction, we tended to find a lot of large corals and a lot of fish. We can look using these maps to find other places similar to that, and we can predict that these habitats will occur there as well. And then we go back with the MARE's Beagle ROV, and we take some pictures to verify that and get some samples to bring back to the lab. So we've learned there's just a whole world of diversity down there, and it's a sensitive habitat, right? Very sensitive. It's not the kind of thing that you can take for granted. So it's important that we need to know where these are, be able to share that information. This is all public. You know, we put our data in we have a database where people can go and see where they are in case they want to sample, or incorporate them into fishing maps, you know, and identify them as a snag or a fishing hazard.
Well, this is something that you know, obviously you've noticed the same thing since we started this work. Yeah, the Channel Islands really didn't have a sense of where any of that habitat was. You had some pretty coarse bathymetry maps, which would just tell you that there was a big, deep depression, but really what it was constructed of in terms of rock or just mud slope wasn't available. And so I know that over the last, 5 years, that the mapping around the Channel Islands has really allowed for that development of, like you said, the model-based approach to really expanding out what we know, because we now have that mapping. And so I think that's showing that sort of technological side of things, where the technology to go out and actually get these high resolution maps becoming more available, combined with, you know, being able to go down and actually visually survey what's in those areas and then come back and model that is like you said, helping to put forward those things. And then again, like EFH's essential fish habitats, as you will, that are protected, it's driven by your work and our work together. So that's good. That's a very good point about the Channel Islands. I know that the Channel Islands became another interesting spot for, you know, climate change. We're still always looking for that sort of that signature. If anything that you've gleaned from any of the work that we've done so far?
Yeah, yeah, absolutely. One thing at the top of the list is just it's a great place to work. There aren't a lot of places you can get from shore to the deep sea in 4 hours. You know, out
Right.
here on the East Coast, we got to go overnight, even get into deep water. So, you know, it's got this cold, highly productive water, this dramatic topography, and some of the features there go all the way down from 300 meters, so about 900 feet, all the way up to about 40 meters, right? And
Hmm.
one of those we call the footprint ridge, and it's right next to Piggy Bank, which is
Beautiful
this,
spot.
yeah, really great, covered with corals and sponges, lots of fish, and we were able to deploy temperature loggers there. So usually when we go out, we just take an instantaneous measure of the temperature. Okay, well, it was July and you know it was 12 degrees, but we didn't know what's happening in January or February. But deploying these loggers, we got a record every 5 minutes or so. And what we learned is, as expected, in the deep sea, temperatures are really stable. As expected in the shallower waters, the temperatures are more dynamic. But where we really opened our eyes, I think, was in this middle range. We would see these excursions, these heat waves that would last a week or so. So thanks to the MARE team and the ROV Beagle, we were able to collect about a dozen small Gorgonian colonies, bring them back here alive to our lab in Charleston, South Carolina, and actually, you know, subject them to different temperatures for different, you know, time periods. And what we learned is that excessive temperatures for only 4 or 5 days are enough to cause permanent damage and that they won't recover. What we also learned is it doesn't get quite that hot down where we're seeing the animals, right? If it did, we probably wouldn't be seeing them there. But we are close. There is a threshold. We know what that threshold now is now. It's around, if I recall, it was like around 12 and a half degrees or maybe 15. But if it gets that warm for too long down in the deep sea, we would expect long lasting damage, right? So now we know that threshold, we know how to measure it. We've still got loggers out there. So if we were to see some warming event that lasted unusually long, we would be able to go back and verify whether the threshold holds up in the deep sea. But fortunately, the corals that are down there seem to be doing okay so far, and we don't reach these temperatures.
Well, good. And then this is where all, you know, monitoring and being able to go out there regularly to, you know, to check this kind of thing, just to make sure that, you know, what you're learning in the lab does apply to actual field applications. So no, that's great. Well, that actually sort of moves us into the other work that you do. If we jump now all the way to the Gulf, do you want to talk to us a little bit about the projects you have going in the Gulf following that Deepwater Horizon spill?
Yeah, sure enough. So I started at NOAA back in 2010. And I don't know if you recall, but that's when the Deepwater Horizon oil spill happened. And I remember standing in the airport looking at the TV, and they're talking about it's in the deep sea and they can't stop the oil spill. That's when I realized, oh my gosh, this is about to become my job, right? That's what I was hired to do. And I did my PhD in the Gulf of Mexico there at Texas A&M. So we knew there were corals down there. And we knew where they were. There were some experiments already in place. So as in April was the spill, I think we got in there 6 months later and checked out the injuries. The first teams went down to the very deep sea where the wellhead was actually gushing out of the seafloor at 1,500 meters. And they were noticing this plume that was above the seafloor and impacting the seafloor. And when they went to look at the corals, they had suffered some pretty severe injury. They took samples. They were able to fingerprint the oil that was on the corals and match it back to the Macondo well 252. So that was very important and fundamental work. Then we realized that, but wait, there's also all this oil on the surface and this huge slick that's visible from space, right? So because the animals are feeding on this surface plankton, we figured there's got to be a connection. Basically, what's happening is we have oil and dispersants being applied onto the plankton that's then raining down in these mesophotic reefs, which are about 50 to 100 meters deep, where we work a lot in the Channel Islands, that same depth zone. So when we went down and looked at those rocky reefs, we did see about half of the sea fans had suffered some level of injury. And we compared this to pre-spill conditions, and we found that more like 5–10% of the colonies had some level of injury. And our work out in the Channel Islands has confirmed this, that we expect maybe 1 in 10 or 1 in 20 colonies to have some kind of overgrowth or maybe some tissue loss. But there is, we have used Channel Islands. It's like always to check our assumptions, If we see this here, we go look there. Yep, it's there too. So that's good to know. But now's come the time for restoration. All this science was put together, and we got the largest environmental settlement ever, $21 billion from British Petroleum Company. And there's a 15-year duration to the restoration work, and we're in year 1 right now. So we're starting to plan our activities. Those activities will, involve revisiting the sites where the oil spill occurred, see if these colonies have improved in condition or they've declined in condition. It could go either way, right? But we're hoping they recovered. We take heart in the fact that about half the colonies were still okay. There were some young recruits there, some small corals that we expect would grow back. But in order to restore these reefs directly, we have to propagate these deep sea corals in the lab and then transplant them back in the deep sea. The other way to do it is to put down some structures, like some artificial reefs that might recruit new corals. We've documented about 100 to 200 corals in 3 different species that we know were injured. So our short term, and by short term, I mean for the next 7 years, we're going to try and directly compensate the injury that was done by the Deepwater Horizon. And the next 7 years after that, we're going to try and scale that up. And, we'll be working with a broad group of partners, academic experts, different ROV teams like MARE, different kinds of projects involving mapping, modeling, habitat assessment. We're going to try and directly restore these reefs, but also protect ones that are similar, like Flower Garden Bank National Marine Sanctuary. They've got similar aggregations, and then we'll use the models to find new ones that we didn't even know about. So we have an approach for direct restoration and then an approach for mitigation. And that work's just starting now. And frankly, I just want to thank you, Andy, and the whole MARE team, because if we hadn't have had these opportunities to collect the corals, keep them alive, and test, some of these exposures of temperature and ocean acidification, we wouldn't be set up to do the work the way we are now. So it's been a great and very fruitful collaboration.
Oh no, it's been, for us, it's so educational. I came into this knowing a lot about shallow water species, but as the years have gone on, my understanding now of deep water species, and it just sort of opened my eyes to their importance and their sensitivities. And so that's an interesting, so at the Deepwater Horizon, you were able to then, I guess, sort of litigate a little bit for the restoration aspect. What was the driving force behind that? Obviously, you said you could show injury, but you had to have then something to compare to. And so that baseline, I think, is what you were referring to.
Absolutely. Absolutely. And you don't have that everywhere. The Gulf of Mexico is U.S. major industrial basin, right? We have oil and gas, we have major fisheries, for shrimp coming out of the Gulf of Mexico and oysters. So there are areas where we do have lots of pre-spill surveys, definitely for snapper and grouper, right? The fisheries departments have been looking for those kind of fish and new reefs to manage or to know about. And, you know, we couldn't have really made the assessment without knowing that baseline and not knowing how to get that baseline. Now in Channel Islands, I mean, come on, you guys got 4 oil rigs right outside the sanctuary boundaries. I don't think they're operational anymore now.
No, they haven't been active for a while, yeah.
Right, but you had that Refugio oil spill. Remember that? Was that like 4 years ago or something?
I think so. I think that was when they shut the last ones down, yeah.
Yeah, so now we know that these corals are, we call them sentinel species, right? You know, it's hard to go out and count the fish because they move around a lot. But the corals, they're pretty much going to stay put, we hope. And when they, you know, are exposed to oil, exposed to pollutants like pesticides, or even, you know, the dispersant, which is probably the real problem in the Gulf of Mexico. Our experiments have shown that the dispersant is just highly toxic. They're somewhat adapted to the oil on its own and have mechanisms to defend some amount of that. But, you know, there's a whole world of chemicals that can cause injury down there, and we need those canaries in the coal mine. You know, we need to know how to observe the injury and how to document. But now we have a great baseline in Channel Islands, and we know, you know, where they are, what they look like a couple of years ago. And so in the event of, you know, God forbid, a future disaster, we'll have that kind of information in order to do that assessment.
Well, and that sounds, you know, it brings to the next kind of point. Then protecting deep sea corals, it's really is important. If you're saying then they are the canary in the coal mine, is that seem correct then, as far as a bottom species?
Well, they're definitely highly sensitive to temperature changes and acidification, depending on the species. I see Natasha's got a question for us.
I see that too.
Which is worse, acidification or temperature rise? Okay, well, it depends which coral we're talking about. If it's a stony coral, like this Lophelia, acidification is a big problem, but it also can't survive above like 15 degrees C. For the Gorgonian corals, they seem to be the winners in an ocean acidification scenario. Their axis is it's protein. It's kind of like a woody stem. When we collect them, and you can break them like a twig, and you can see that there's a little calcite in there, but that's not the problem. For the Gorgonians, it's the temperature sensitivity. One thing we don't know about at all is the food. You know, both of these conditions will change the food source, change the phytoplankton, and that's something we need to learn about. Is does a change in the surface plankton affect the health of the communities down below?
Yeah, and I know that you know, just from having conversations with people, you know, aquariumists and things like that, that some of these coral species are very specific on the type of food source that they would go for. So a complete change in, like you said, that phytoplankton may affect their ability to feed on the specific organism they're looking for. Does that seem correct?
Yes, absolutely.
Okay, well, it sounds like we've covered a lot. Obviously, protecting deep sea corals has been a big push. Just if we could close it up with, how are ROVs helping with that effort? Obviously, we go down, we collect them, we get video, but what else is it that, I guess, is helping with your efforts?
Well, what helps a lot is that we can get the samples we need very delicately without causing, you know, injury the way a dredge or a trawl might sample the seafloor. And the Smithsonian Museum is full of dredged and trawled samples. And the problem there is you get the sample, but you never know what it looked like alive, right?
Right.
you know what it looks like on deck and in ethanol. But the ROVs give us a chance to, first of all, observe the animal alive in its natural habitat, right? So we see the corals, then we see all the animals associated with it. We're not going to be able to collect all those animals, but we'll get a photo documentation or a video documentation. And then when we collect that sample, as you know, we usually just only need a little piece. We don't have to get the whole coral unless we're trying to assess its age, right? Which we only need to do once in a blue moon. But we can get those little samples, which are enough for genetics, enough for taxonomy, enough so that we can put a name on this purple thing that we're looking at.
Looks like the other one, right?
That's right. So the ROVs are invaluable, and they're also giving us a whole new perspective on the seafloor. Not a lot of people know this, but most of the seafloor biology comes from these down-looking cameras that were towed over the surface. So you see a lot of crabs, you see a lot of sea cucumbers and stuff, but you don't get a sense of what's happening in the water column or with the upright animals. And because the ROV has this forward-looking camera and you're so low to seafloor, I think we're usually flying, what is it, about a half a meter?
Yeah, about a half a meter.
Yeah, so we're real close to the seafloor, getting, you know, the benthic view. And this gives us a whole new perspective on that kind of habitat that we didn't have before.
Yeah, no, it's been a interesting discovery for me too. Is just that when you get down there, things that I've seen in like you said, other perspectives, the downward looking, where you go across it and you kind of saw a pink smudge, but you're not really sure what it was. And then to be able to actually stop and hold on it and film it and see something interact with it, it definitely is a different perspective and allows you, I guess, a little bit more depth in what you're gaining from that same imagery.
Yeah, yeah. And plus you've got your GPS and your digital positioning. So we know within a meter or two where that coral actually was. You can't get that kind of information from a trawl or a dredge or even an AUV, a robot that doesn't have a tether. They're estimating location based on, you know, physics and water column characteristics. So, you know, it's important for us to be precise, especially if we're going to manage these areas. We want to make sure that if we're putting something off limits, we know exactly where that is and that we're not just guessing.
No, agreed. It looks like Natasha had asked one other question about the baseline pictures. Who's most interested? Would that be local governments, state, national governments, industry? It let you feel that. It seems like everybody would be.
Yeah, well, these pictures are useful for a lot of things. I mean, everything from posters and brochures to actual scientific data. We use these images to measure the size and health of these colonies and count how many brittle stars are on it. But the person most interested, other than the scientists, will be whoever has jurisdiction, right? So state waters go out to, what is it, 3 miles.
Yeah.
Right. So, and you've got deep water there within 3 miles. And then beyond that, it becomes a federal jurisdiction. And beyond 200 miles, we're into the, you know, the high seas. And there, nobody has jurisdiction.
Right.
They can try, but you got to have that evidence. And the photos are more and more, you know, I'd rather have 1,000 photos of a habitat than, you know, a half a dozen samples of the things. Ideally, you get both, but the images are valuable. They're a great way to communicate with people. They turned out to be evidence in the Deepwater Horizon case, right? So right. This is how you make your claim.
Well, and you continue. You plan to continue this coral work into the future, looking at obviously, you know, the areas that we continue to monitor and you know, still learn about, but also expansion into new areas. Is that?
Yeah, for sure. I mean, we want to know how they reproduce. We know their names now. We know how fast or slow they grow, but we don't know exactly how they reproduce. And if you know from shallow coral habitats, once you know how they reproduce, you can actually get within 5 minutes of their actual spawning time at places like flower garden banks. And this is based on the lunar cycle, changes in temperature. So it's usually around August, and they can dial that in. But we have none of that information for these deep water animals. We just have snippets of info here and there. So we want to start monitoring these more regularly, figure out how they reproduce. Right now, we reproduce them by fragmenting them. So it's asexual reproduction. And what you're going to get is like one gene line, right? That's all you got. And you want genetic diversity, right? So we'd prefer there be some kind of sexual reproduction, but they can also, they can brood larvae. And there's a number of ways these corals can reproduce. So we got to learn that much. It's critical, especially if we're going to propagate them and start transplanting them back in the deep sea.
Well, and I know they're doing that in the shallows, and there's, obviously when you have these die-offs that we saw, obviously in the Gulf where we had a clear sort of, event that you could pinpoint to. We've seen die-offs off of California where, it's not really clear as to what, you suspect it might be temperature related, but it's still unclear. But obviously then you have this, as you said, sort of cloning of corals, but you imagine that then there would be some sort of combination of cloning and then trying to brood or create starter populations, that are maybe genetically different in close proximity to try to, get that genetic diversity to, spread out from there.
Yeah, that's right. And, that takes a lot of samples in order to figure out what's that genetic diversity. Already we've seen that although the animals are pretty broadly distributed all the way from the Caribbean up to off Cape Hatteras, that there's a couple, we call them metapopulations. There's different genotypes in different regions. And we wouldn't want to put the wrong genotype back in the wrong place. So we have to understand that stuff before we really start doing the work in earnest.
Well, it sounds like you've got a lot of work ahead of you. It sounds like you got a 14-year plan. So most people are having trouble with the 1-year plan.
That's right. We're hoping we can suck you guys into some of this work. I know life is good out west, but
we're always happy to join you. It's been, like I said, for me, just the amount of information that I've learned from you and working with other researchers on these projects is, it's why I got into this as well. It's sort of the unknown and knowing about it, and so it's been a real journey. Before we bring Natasha back, what one thing, if you could, I guess say anything to people, what one thing could people do to help have a positive effect on the ocean? That is a little thing that they could do each day that might help a deep sea coral even.
Yeah, that's a good question. Most of the things that come to mind for me are pretty much in the grocery store. The decisions you make there, whether you're gonna, have paper packaging or plastic packaging. Here in Charleston, we've banned the plastic bag. So it's all paper bags, and it made a huge difference. That was like a year and a half ago. Nobody even remembers that we had plastic bags before. Right? It was an easy change, but you don't see that everywhere. We went up to New York; they're still dishing out plastic bags, and you see them in the trees. You see them out in the road. Yeah, and so that's one thing. Also, know your seafood and its origins, and choose sustainable seafood options, and preferably, get that seafood locally if you can, rather than, shipped overseas. But one thing I try and tell people, especially my students, that I think the best thing we all can do is get out on the water and enjoy it and remember why we're there trying to protect it, and then, share that experience with our friends and family.
Yeah, no, that's and something that you said earlier today, talking about some of the chemicals that were in the water. It's sort of maybe remember that, even like you said, the food that we eat, and there's a chemical side to that aspect of it. Organic maybe less, the things that we put down our sink. A lot of this ends up back in our oceans. And so keeping in mind those things as well, like you pointed out, chemicals can have a big effect as well.
Yeah, and there are organizations that monitor the water quality. I think NRDA is one of those groups, and maybe some others. But this is something that all of us can do, is to support the nonprofit organizations like your own that are out there trying to make a difference every day. This is an easy one. We tip our waiters and our bartenders. I think we should also be donating to folks like yourself that are really making it your mission to make sure our oceans are clean and safe to swim in.
Well, thank you. And thank you for coming on today, giving the interview. And look forward to what we do next, getting back out on the water and discovering something new. So thank you again.
Yeah.
So we can bring Natasha back.
Peter, what a perfect segue to wrap up our session. Thank you, Andy and Peter. This was great. Deep sea corals are amazing. I think the first time it kind of hit me was realizing that these corals live below the photic zone, and they are not like their shallow water cousins. These are animals. And people don't, they look at corals and it's kind of mind blowing that those are little animals in there. Yeah. So I know that blew my mind when I started working with MARE. And I hope we got everyone excited about deep sea corals. They're not the the marine megafauna. They don't get much love these days, but they are the forests of our ocean. And without them, the rest of it doesn't work. So they're critical. And I'm so excited about the work that Peter's doing. I'm so excited about the work that MARE gets to continue to work with you on, Peter.
Yes.
Is there anything else you want to tell us about deep sea corals before we move on to a couple movies?
No, I just want to say thanks for having me.
Okay, great. And I think, which is one of the things that's so critical is if we don't document and measure what's going on, we can't protect it. And what you were talking about around the Deepwater Horizon spill, if we hadn't had that information before, we wouldn't understand that impact. And that is why MARE's work is so critical, because if we don't go out and document and measure what's out there, how will we understand the impacts, whether it's fishing, climate change, ocean acidification, all of the above? So I think, yeah, that's a great example of that.
Essential information.
Yes. Thank you to all of our viewers. We got some great questions. Keep the donations coming in. Deep sea corals need love too. And we're going to show a couple videos now. So stay on. Thank you again, Andy and Peter. And we will show some videos and then have our next session at 3 o'clock with Lance Morgan from the Marine Conservation Institute talking about blue parks. If you want to know what a blue park is, come back at 3. But stay tuned for octopus and deep sea coral videos that we've been talking about.